Welcome to this next episode in our Emmes at Home series, a program developed by Emmes to share thought leadership safely and securely from their homes during a pandemic. We are pleased to welcome our moderator, Jill Barrett, and her colleagues in the Vaccines and Infectious Disease Therapeutic Research Unit at Emmes.
Hi, and welcome. I'm Jill Barrett, a project leader in the Vaccine and Infectious Diseases Therapeutic Research Unit, or VID-TRU, at Emmes. I currently am the director of a project studying more than 55 different infectious diseases, and I personally have worked on clinical trials for pandemic influenza, Zika, and coronavirus, as well as some other pathogens over the last 16 years. I'm joined today by a panel of my Emmes colleagues, each of whom has extensive experience conducting clinical research during infectious disease outbreaks and pandemics. What makes our discussion today so unique and interesting is that we're not just talking about conducting clinical trials against the backdrop of COVID-19. We're going to talk specifically about our panel's experiences supporting clinical trials that are actually targeting the pathogens causing these endemic or pandemic outbreaks, such as Ebola, Zika, or now COVID-19. So without further ado, I'd like to introduce our panel. Phyllis Renahan is a senior project leader at Emmes with over 25 years of data and project management experience in infectious disease clinical trials, primarily supporting Emmes's work with the Vaccine Research Center. A recipient of the NIAID Merit Award, Phyllis made significant contributions to the Ebola outbreak response and clinical development of a promising vaccine candidate. Phyllis also led a large international Phase III trial of a Zika vaccine candidate throughout Central and South America. Dr. Matt Mikowski is a senior biostatistician with Emmes. His research has pioneered new paths in modeling high-throughput sequencing data. Matt has supported vaccine studies for a variety of pathogens within the VID-TRU, but he's spent most of his time since February 2020 supporting Emmes's COVID-19 vaccine and treatment clinical trials. Dr. Carrie Petrie is a project leader at Emmes with over 15 years of infectious disease experience. Carrie has been the program director for Emmes's IDIQ contract with BARDA since 2015. And in this role, she supported the STRIVE trial of Merck's Ebola vaccine in Sierra Leone. Carrie has worked on studies for a wide array of infectious diseases and has extensive experience supporting pandemic influenza and universal influenza vaccines. And lastly, Jessie Kennedy joins us as an associate project leader at Emmes and standards manager within the VID-TRU. Jessie manages a portfolio of 15 to 20 active studies in tropical and parasitic diseases such as malaria, including many international studies. As standards manager, Jessie brings expertise with VID's large library of standard materials to support infectious disease studies. Thank you everyone for joining us today. Let's jump in. So obviously, vaccine and treatment studies during a pandemic are highly urgent and speed is critical for public health. So how have you been able to accelerate study startup timelines and rapidly execute studies in this environment? Who wants to get us started?
One of the advantages that we have in the infectious diseases therapeutic research unit at Emmes is a large pool of dedicated staff who have worked on a variety of infectious disease trials in the past. And we share our resources across the research unit. So when we get a large scale epidemic or pandemic trial that we need to respond to rapidly, we have a good set resource pool from which to draw experienced staff to support the trial.
Great. Jessie, do you want to chime in with how our standards efforts have helped with our response?
Absolutely. And I also would like to highlight our data system, Advantage eClinical. It's a proprietary electronic data capture system that we have been refining for many years, and particularly in the context of infectious disease studies, have developed a wide array of standard electronic case report form templates. These templates are adaptable to a wide range of studies within the infectious disease TRU, including preventative types of applications, therapeutics, and of course vaccines. this library of eCRF templates allows us to put up our data system very quickly upon request from a client.
Matt, as a biostatistician, how can you talk to the tight timelines and also and not just writing and setting up a study, but responding to the analyses of the study as well?
So I can start by just piggybacking a little bit on Jessie and kind of some of the standardization that we have. As she mentioned, we can set some of the systems up in as quickly as 48 hours. And part of that allowed us, for example, the randomization, which we have kind of standardized randomization plans that can be reused, some standard coding that can be used. So we can set these up pretty quickly. And similarly, from writing a SAP standpoint, we have, again, standardized, so which allows us to pull in multiple people and kind of work in parallel to get things up very quickly.
So I'll build on that a little bit for the STRIVE Ebola vaccine trial in Sierra Leone that was sponsored by the CDC and conducted under our BARDA IDIQ contract. We also relied on many of those same standards that Jessie and Matt just described. Sierra Leone was a challenging environment. They had just experienced the worst of the Ebola pandemic when this trial started. There was limited clinical research experience in Sierra Leone. Much of the healthcare population was directly impacted by the epidemic, and so many trained clinical staff were either not available or not in the area when we were conducting these trials. And those standards that we had developed allowed us to train the staff that were working on the trial, many of whom were medical students or nursing students or pharmaceutical students and did not have a lot of research experience. But we had such great guidance from our standards group. We also relied on the adaptability of the data system to create the data system very rapidly for that trial. And probably most importantly, we were able to travel to Sierra Leone several times over the course of that study and interact directly with the people who were doing the medical and safety assessments, as well as collecting and entering the data into our systems. And we could use the training deployments of Advantage eClinical to assist in the training. And that was also incredibly helpful. On some of these trials, for the STRIVE trial, we also worked with other CROs that we would normally be competing with. And we all had to work together. It was actually very inspiring. Everybody came together to work towards the common, very important public health goal of developing the vaccine. I think one thing that was really critical for the success of the entire trial, and especially for the components that Emmes contributed to, was that we had a very stable core team over the entire 2 to 3 year length of that trial. So the same medical officers, the same statistician, data manager, project manager. And while staff turnover was quite high, especially within the CDC and some of the other CROs, we were able to maintain that historical knowledge of how the trial had been conducted from the very beginning, the reason why analysis decisions or data collection decisions or safety monitoring decisions were made. And that just proved to be really invaluable, leading all the way up to licensure of the vaccine by Merck and supporting Merck in submitting their data for licensure.
So as we all know, in this year, everything changes day to day, week to week, and these pandemic trials are no different. So pandemic trials often require frequent changes and adaptations as we learn more about the disease. So Matt, where have you seen the greatest need for flexibility, and how have you handled those sudden changes and your support of the COVID studies?
So I think kind of one of the main points is, as you said, as we get more information about the disease, we may need to rethink how we're planning to analyze the trial. So especially in our COVID trial ACT, we had to think about when we would analyze our final endpoint. Originally, we had considered day 15 as when we would potentially see an efficacy effect, but as more information came in from China and other studies around the world, it looked like the disease progression may be a little longer, and day 15 may not have been appropriate. So we just had to rethink it. One thing that allowed us to do it, because this was at a point where we had already started enrolling, is having separate blinded and unblinded teams. So in order to keep the trial integrity, only the blinded team was kind of in on these discussions and made final decisions, whereas anyone that was already unblinded to data would not be part of any of this decision making. And this allowed us to change our primary endpoint prior to final analysis to kind of use something that we felt was much more appropriate moving forward. Another point that where you require flexibility is also just dissemination of information. As we used other trials' information to change our endpoint, we also want to do the same thing, is get kind of our information out as quickly as possible. And one place where we saw it is at the end of Act 1, where we had a DSMB report, a DSMB meeting at our, what we would say our 100% information fraction. And the endpoint was significant there. We still had some follow-up to do, but at that point, that was our own possible treatment that was going on during the pandemic. It was still relatively early. So basically, the DSMB met on a Monday, recommended unblinding. The next day, we did another freeze, ran a few tables that kind of we were ready to output, and Dr. Fauci was on his way to the White House to get this information out to the public. So kind of the kind of flexibility, just all of a sudden on the dime, just being ready to provide information out to the public. And we're starting to see actually a similar thing coming out of our part 3 of this trial as well, some safety concerns, and just collaborating with the FDA on what is the best way to get this out to the general kind of treatment groups, so that they know how to treat their patients and get them the best help possible.
Right, because we're still in the midst of this. We need to share the knowledge as we're gathering it. So if you find out that a certain treatment is a safety risk in a certain group of people, that's very important information to share, even if we haven't gotten to database lock. So thanks.
Correct. Yep. And just to add, it's also just kind of going back one way or the other, because these trials, in a pandemic, these trials are very highly scrutinized. So you get a lot of criticism for kind of whichever decision you make. So you also want to be careful that you don't go too far in the other direction of, if people are saying you gave this information too early, don't necessarily wait longer later. Just trying to find that proper balance as well is important in these trials.
Right. So I know that we are often also urged to follow up those releases quickly with a peer-reviewed journal article and sharing our data. And I know for the Act 1, that announcement from Tony Fauci the day after the DSMB meeting was very exciting, but then immediately put us on the clock for getting the results to FDA, getting the New England Journal of Medicine article published, getting all of those ducks in a row while we're still doing follow-up and trying to lock the database at the same time. So the compression of all of the timelines, everything together during these pandemics is really unique. It's so important and really highlights our flexibility in being able to meet those. Carrie, did you have anything you wanted to add?
Yeah, I think maybe just kind of another aspect of this, kind of almost in the opposite direction of where Matt is going with the ACT trials, but for STRIVE, due to considerations of the local population on the ground and the regulatory discussions happening in Sierra Leone, they were not able to start that trial until the epidemic was ending. And so by the time they began enrolling and vaccinating individuals in the STRIVE trial, there were relatively few cases in Sierra Leone. And STRIVE was an efficacy trial, so that really did impact the type of study that could be done. We also had to be flexible for that study, and ultimately we were able to produce an enormous data set of safety data, including memory aid and vaccine reactogenicity data, which is challenging enough to collect in the United States, let alone in Sierra Leone. And that data was, again, critical to the licensure of the vaccine. And so we have that experience to be flexible within our statistical group and the data management group to really adapt to the changing conditions.
And Phyllis, you had a similar experience too.
Yes, so for the Zika trial that was conducted in Central and South America, similar to what Carrie described for the BARDA trial, by the time that we were able to get all of the sites up and running in the trial, for the most part, the incidence of Zika had kind of waned. And so similarly, we're in a situation where we're trying to conduct an efficacy trial, and we're in a position now where really all that we can speak to are the safety profile of the vaccine, which is still very important. But you have to really take those types of things into consideration, particularly with international sites that have lots of additional regulatory obstacles to be need to be addressed before the trial can actually be initiated in those locations, and making sure that you're able to do that as rapidly and efficiently as possible so that you are able to enroll the study population during the course of the pandemic and not after the number of cases has waned off significantly. So that will definitely has an impact on the outcomes of the trial.
And I think we learned a lot as a scientific community from both Ebola and from Zika. I know at the last NIAID council meeting, they were talking about reflecting back on the Zika trials and how can we expedite our rapid response. And we did that. It was a success, essentially, for COVID. It got much faster. But it was also a pandemic, right? We could start the trial in the US because we were affected in the US. We weren't trying to get regulatory approval in Sierra Leone or in Brazil or around the world. So I think that together we're both learning from the experiences, but also just being aware of where the delays might be prepares us for the future as well.
I think just also adding to the vaccine trials with the COVID, the response was definitely much quicker, especially with the mRNA vaccine that we're part of here at Emmes. Is just the sequencing and then straight to kind of vaccine was extremely quick. I think it was like 30 days or something. And then just kind of starting to run things in parallel and get things out there, especially with this phase I, we were able to use data to help justify going pretty quickly into the phase III trial, including this. Some of the flexibility that we could do is originally the phase I was just for healthy subjects 18 to 55, and rightfully so. It was pointed out, well, most of the people that are affected and will get the vaccine first are older subjects, since they're the higher risk. We were very quickly able to add, update the protocol, update SAP. Add in cohorts for older subjects, able to enroll them, vaccinate them, and start getting that information out there as well that could be used for the efficacy trial. And kind of similarly, as Jill said, the U.S. is affected by these. For a phase I trial, you're actually looking at areas where there isn't too high of an incidence, because you want these healthy subjects that aren't affected by it yet. And kind of our original site turned into a hotspot. So another piece of flexibility is, we were forced to kind of very quickly add a few extra sites that weren't necessarily in areas of hotspots to try to keep that risk of people being infected by COVID that are in this phase I trial limited.
So one of the more remarkable achievements has been our expedited startup of the COVID-19 treatment study, our ACT study. So Matt, can you kind of walk us through those first couple of weeks and how we got it up and running so quickly?
So we basically got the go-ahead very suddenly when it was announced that the cruise ship was coming back and we had COVID patients. So we wanted to be able to get Remdesivir to them as quickly as possible. And one of the ways that we've been able to do this is, as we've talked about before, is some of our standardization. So we basically did it in a, I'm going to call it a tiered setup, where we started designing our system with our randomization and our enrollment forms so that we could get these patients enrolled and get drug to them. And then kind of moved on to add the rest of the forms. And we were able to do that by, designing all these paper forms. And as we said before, so this way the sites could collect the information on paper forms, even if they did have to take pictures of them from the rooms because they weren't able to leave the rooms of infected patients once the form was in there. So, but basically, going back, that's how we were able to do it is we first did all the enrollment so that we could enroll the subjects, and then in stages add in system while still having all the paper forms ready to go and be able to collect the data. And also, we had these cruise ship patients that were the first ones, and then we had a little bit of extra time in between before enrollment fully ramped up. But once it did, it just went crazy as well, where we were most people in the first month to 6 weeks.
Yeah, and I remember I was pulled in to be part of that initial startup. I got a text from my boss on a Tuesday evening saying, "We're doing it. Let's go." I'm going to start building the screens right now, building the enrollment screens. Can somebody QC them? We worked through, so that was Tuesday evening. We worked through getting all of the sites set up. We initially set up only 50 sites, where we later expanded that to 75 and then 150 just in case. And then we had that system in production ready to enroll by Thursday night. So within 48 hours, the site initiation visit was the next morning. We trained the site on Friday morning. That site was activated Friday afternoon. And we were at happy hour Friday evening, back in the days when we still had happy hour. And we got the text or the email that came across while we were at happy hour that the first subject had been dosed with vaccine, or not vaccine, with the treatment. So that was extremely exciting, but just the beginning, like you were saying, Matt. We still had lots of work to do. We continued to get the work on the ECRFs, the MOP, all of the instruction documents, the reporting, the SAP, get all of that ready. We had the data system with all of the ECRFs ready the following week, where we were able to start capturing all of the clinical data directly into the system or from the medical record. So those were the early days of the pandemic. I believe it was February, which seems like 10 years ago. But even last month, when we might have been earlier this month, when we locked the database it was earlier this month, beginning of September, we locked the database for ACTT. We locked the database on Thursday morning at 11 a.m. and our statistical team worked through the night and got the top line results out for that study and over to DMID by Friday evening. So within 2 days. So that our team certainly understands the urgency of these trials, the importance of these trials, and they take it very personally to do their best and do whatever they can to contribute to this solution. So we talked a lot about how a lot of these trials are international. With COVID, we have both domestic sites and international sites, but a lot of these studies that we do in endemic areas or these epidemic outbreaks are international. So what are the challenges that we face with international studies, just typically, but also in the, with the backdrop of a pandemic?
Well, we spoke a little bit about that earlier with the regulatory obstacles specifically. Mm-hmm. One of the big challenges we had for the Zika trial was just cold chain management of research samples and study product. being shipped into the country and then out of for the study product, and then to get this research specimens shipped out of the country. And there were certain situations where study product was stuck at a depot for a longer than anticipated amount of time because the import-export permits were in place appropriately, or some other challenge that was local to the individual country, and study product wasn't usable because of it. We've had situations where research specimens similarly got stuck at depots trying to be shipped out of country. And unfortunately, those samples were compromised, and we're not able to use those samples. One of the ways that we mitigate that specific risk is by ensuring that when we're sending shipments of specimens, that we're not sending all of the aliquots of the specimens in a single physical shipment. We send half of the samples in one physical shipment and only instruct the sites to send the second set of specimens once they have received confirmation that the first set of samples were received at their destination in good shape. But unfortunately, there are situations where samples were lost and where study product needed to be replaced. So those types of things posed greater than normal level of anxiety and issues in certain locations, particularly in Brazil, another location where a lot of the regulatory issues were found. It's a lot more. There's a lot more involved in getting regulatory approvals in certain countries and getting the import and export permits actually established to make the import and export of materials related to the trials more as efficient as possible. And sometimes that's just not possible.
So Jesse, I know you work on a lot of international studies and have for years. So what are some tips and tricks that you can share about how to get around these logistical issues?
So as you can imagine, clinical trials are never without logistical issues and problems, but particularly in international settings, we have experienced many unexpecteds. An example of one of these unexpected situations, it can be, as Phyllis mentioned, aside from the regulatory and logistical issues of shipping study product and clearing customs through many different countries, we've also experienced issues even with things as minor as the types of labels that we put on tubes. A lot of these studies are being done in very hot, humid climates, and it's not always conducive to the types of labels that we're used to in our more domestic settings. So even having to think through the type of label that we would place on tubes can be challenging, and getting the materials to the study sites. Another example we've encountered has to do with the climate at which these studies are taking place. We've had many issues and challenges with specimens being lost due to power outages. These are resource poor areas. There are unexpected coups or political situations that impact the ability of the sites to monitor the storage of samples. And on several occasions, it's resulted in freezers not having power and those samples being unusable. So it's just thinking ahead with a wide variety of unexpected situations and kind of planning and putting into your procedures safeguards to minimize the risks if and when these types of situations do occur.
I also remembered while we were talking that we had a Zika study in Puerto Rico, and so it's hard enough that it was Zika in Puerto Rico, but then Hurricane Maria came through, and the clinical site had a lot of damage, and we lost a lot of specimens due to power outages. And so I guess, have you learned, some best practices to avoid this, to prepare for it? I would think, kind of get those specimens to a safe place as fast as they can, or what are some things that you've learned from that?
One of the things is definitely requiring that the international sites have backup generators, backup power supplies on site. So if you're working with a site management entity that's helping to coordinate the trial from the site's perspective, making sure for certain that they have a backup generator in place, and temperature monitoring on their freezers, so that they're constantly recording the temperature of samples as well as study product. But definitely the backup power supply is probably the most important thing specific to, at least to this research specimens in the study product.
Yeah. Carrie, can you speak to any situations about collecting specimens in an area where there's an outbreak like Ebola in West Africa, and getting those specimens tested? Do you just put those on a plane and send them back to the U.S.? I imagine not. I imagine it's a little bit more complicated. So how do we deal with that situation?
Yeah, it really was quite complicated, because rightfully so, there are a lot of protections in place on materials being collected from human subjects leaving the country of origin and going to another country for testing. So again, speaking to the kind of regulatory approval process, and we actually were able to work with CDC and BARDA staff who would be going to the regulatory agencies' offices nearly every day to give them updates and share the plans for how those specimens would be transferred out. Part of eClinical, part of the data system that Emmes uses is a specimen tracking system. And often when we work with groups for the first time who are not familiar with Emmes, they kind of are a little bit hesitant to roll out the specimen tracking system. They don't think it's necessary, or they have mechanisms in place that work just as well. And we were able to convince the sponsor for the STRIVE trial that tracking the specimens in this scenario would be actually quite helpful from the site of collection to the local repository, and then when they were ultimately transferred to the US and the labs. And they, in the end, found that I think quite useful. I say that because we started to get many requests for reports and information from the specimen tracking system, from Global Trace, which is the name of the system, to help them decide which specimens needed to go where for
I think another thing too, speaking particularly to the participants of trials at international settings, that a lot of people from those cultures don't really trust Western medicine. And so they have this inherent built it like caution, if you will, that guarding them, perhaps preventing them from willfully enrolling in clinical trials because they're afraid of what the outcome will be. Are the vaccines that we're administering to them cause them to catch whatever disease it is that that we're trying to prevent. So there's some underlying element of that, of just this general mistrust of Western medicine. On what Carrie was talking about, I think that at least in the trials that I've supported internationally, we set the bar very high in terms of what we expect from the clinical sites. Not only having to adjust our way of thinking and understanding that that they're going to have to go through a certain decision-making process that might be a little bit different than the decision-making process that we go through here in the U.S., but they conduct clinical research at the same level of expectation that we do in the United States. They don't necessarily naturally follow all of the same rigid rules and guidelines, and it requires a lot of extra time and commitment from the CROs who are involved to train the staff to ensure that they really understand what the requirements are, and to work with them to not only to do the things that we need them to do, but for them to understand why we are placing those expectations on them. And that, it just requires a lot more interaction between the CRO and the clinical sites in terms of training and reminding and things like that. So.
Okay.
I would add that sometimes the reverse is true, in that we are dealing with a set of regulations in certain countries that is more stringent in certain aspects.
Right.
We have an example recently of a study being conducted in Brazil where it has been challenging to determine how to incentivize participation, or how to, what types of remuneration can be provided. This study happens to be in children, and even providing candy after the blood draw is viewed as too much of an incentive or pressure to have the child or the family participate. So, just things we don't often think about when we're used to phase 1 clinical trials in the US, where we're monetizing participation, or somewhat quick to reward children for their time in these studies.
Kind of to add to that, to Jesse's point, just some of the different regulations that you might run into in different countries. For example, just data dissemination. So in the US, we have our kind of standard informed consent forms that we can use, which may not be appropriate in another country, where the data use may be stricter. So you kind of have to be careful of how you word things, and you may have to use different informed consents depending on where you're enrolling that particular patient.
One of the things that we found with our trials conducted at the international sites is sometimes the sites, in addition to having to deal with the ill patients, that are enrolled in the trials at their sites, overwhelmed by the large amount of data that they are expected to collect and enter into the data system. Sometimes there are language barriers. They don't necessarily understand the English queries that we are issuing to ask them to resolve. And they're just overwhelmed by the sheer volume of queries and expectations for data. So we've found that just, having conference calls, have the lead data manager who's supporting that particular clinical site get on a phone call with the clinic coordinator on a weekly basis and just talk through the reports, interpret the issues, help facilitate, prioritize what data they should be working on first. What's the list of the most important queries that they should be addressing first, and helping them to understand what the expectations are. We've just found that having more one-on-one interaction, particularly talking to somebody on the phone, and not sending communications and instructions always just through email, but actually having a conversation with people really kind of helps them through it, helps them understand, and just establishing those relationships with them helps make them more engaged and wanting to get the information entered into the data system if they have that relationship with you. So investing in the time to talk to them on an ongoing basis really helped with getting more rapid data entry and better quality data.
When the site staff themselves are seeing a lot of patients every day, and a lot of sick patients, so how do we deal with those interactions, and what is it about a pandemic that might be a little bit different than other studies?
So I think in the STRIVE trial, the subjects getting the vaccine reactogenicity symptoms were potentially overlapping with signs and symptoms of Ebola. And it was really important in that trial to carefully understand when subjects presented as being ill, whether they might have suspected Ebola, which was the efficacy outcome of the study, or if they were having a reaction to the vaccine. Because if they were only having a reaction to the vaccine, no one wanted to send them to an Ebola evaluation center where they might actually get exposed. So part of that response was being very careful about the training. And again, our medical and safety staff did a large amount of training with the clinicians who were not necessarily in the mindset to think of vaccine reactogenicity versus Ebola. They had been working in Ebola. So part of it was providing that training so that, and the materials and the references and the guidelines so that they can make that decision. And then also, again, just having that more of that open mind of what the site staff were going through on a day-to-day basis. For the STRIVE trial, I remember there was a devastating flood in Freetown during the trial. Several of the subjects enrolled in the trial died during that flood. It was really tragic, but also the coordinating center, the data entry center, was damaged in that flood. And so, you know, that's not a situation we necessarily have to deal with frequently for domestic studies or non-pandemic studies. So just, you know, keeping that in mind, the challenges that are being faced on the ground, I think, or in the hospitals or in the clinic sites is important.
It is a challenge to weigh the rigorous standards that we are held to in studies, especially in the US, best clinical practices, FDA standards for data and whatnot, against the more human element and aspect. And I think, Jill, you hit the nail on the head. It's very hard to ask a coordinator to go complete a form when they're seeing patients, you know, in acute situations. They're dying, they're dealing with a lot, and it's even more enhanced in a pandemic.
I think that's the balance is what is key there, because for COVID, we obviously, for these treatment trials, we have to enroll them as quickly as possible, get the subjects, you know, treatment if they need it, if this treatment works. But we need the data too, because we can't evaluate if it's working. We can't share the knowledge with others until we have the data, and the data are clean and we can perform the analyses. And that's our job as the data statistical coordinating center, to care about those things. But for the research nurses, it's their job to enter the data, but it's also their job to care for the subjects. It's very hard.
More positive note, you know, just one thing, not just from the STRIVE trial, but almost every trial we do, is really brings a sense of like gratitude for the staff at the clinics, at the laboratories working on these trials, and the additional activities they do for research beyond their standard of care, but also for the participants. I mean, a pandemic's always a little different, but you know, for any less than healthy patient population that is willing to engage in a clinical trial, it's kind of an amazing extra sacrifice that's really important.
Going back to kind of collecting the data and balancing, kind of showing empathy for the family, especially if subjects are transitioning to comfort care or hospice. You know, they may want to just be done with everything, the trial completely, but it's still important for us as the data coordinating center. We still want the data all the way through death, rather than having them withdraw from the trial, just to be able to analyze everything. So just striking that balance again with empathy towards the family, but also trying to get as much data as possible.
Yeah, I think we've took the same approach for the ACT study, not just international sites, domestic sites as well, because they super busy sites in New York City, and when New York City was a hot spot and 90 subjects enrolled and they needed, you know, help cleaning their data, get on the phone, the data manager would get on the phone with them, just walk them through the queries, walk them through how to resolve it in the data system. It cuts out that back and forth. It gets it clean, quick. And right, and it builds that relationship, which is going back to our relationship building question. It's really key for the success of the trial. Great. That's a great conversation. Anything to add?
So I'm not sure how relevant it is, but I found it interesting, the issue we kind of ran into at the beginning in response to just an infectious, this infectious disease where these patients were in isolation rooms. And a lot of times we ask sites to take a paper form and then enter the data later, but if they're taking a paper form into the room, it's not coming out of there. So it's kind of, how do you get it into an electronic system without being able yeah, exactly. So that was just something I thought was interesting in the kind of infectious isolation room setting.
Yeah, that was our big shift. I think that was an aha moment for a lot of us that were working in those early days of the trial, because the study started, a week after we had gotten the protocol. We didn't have all of the ECRFs ready to go, and said, "Oh, just complete on paper, enter it later." And that coordinator is like, "No, no, we're not. No." So yeah, they did exactly that. They would hold the paper up to the window and have another coordinator take a picture of it through the window, so they could have their source. But that paper never left the room. Jesse, Phyllis, Matt, and Carrie, thank you so much for joining us for this discussion today. Thank you for your insights and perspectives on managing these trials during pandemics and for your endless dedication to these studies. I appreciate your time every day, but I also appreciate your time joining on our conversation today. For our listeners or viewers, if you have any additional thoughts or questions, we encourage you to reach out to Emmes. And we also hope you will join us for another Emmes at Home event. Thank you again for attending, and have a great day.